Good afternoon, ladies and gentlemen. My name is Matt Cartmill, and I want to welcome you all on this webcast of the 8th iteration of the BU Dialogues in Biological Anthropology. This afternoon, we're going to be dealing with a very special and very important set of fossils that were discovered in Ethiopia in 1994, and that the describers and discoverers of these fossils have proclaimed and analyzed as really transformative and revolutionary finds that have in a lot of ways erased what people thought about the earlier stages of human evolution in the early 20th century, in the second half of the 20th century, and gone back to the earlier part. During the first half of the 20th century, there was a general consensus among people who studied human evolution and our relationship to the other primates that human beings never went through a long-armed, ape-like, arm-swinging or brachiating phase in our evolution, that the human lineage had come off quite early. Some people said all the way back in the Oligocene, when the ape ancestry was still largely quadrupedal. And the argument was that if human beings had become long-armed, long-handed, arm-swinging, ape-like animals, that would have shunted us into a very specialized kind of locomotor adaptation from which our ancestors would never have been able to escape into a bipedal kind of short-armed adaptation when they came to the ground. All of this changed in the late 1950s and early 1960s with the advent of what was called the new physical anthropology. And during this phase, the received wisdom switched over to a very different kind, to the opposite kind of assertion, of claim, that in fact the last common ancestor of humans and chimpanzees had been very much like a living chimpanzee, with a few minor differences, both in its ecology, its locomotor behavior, and probably in its social organization. The discovery of the Ardipithecus ramidus fossils and the Ardipithecus 500 skeleton from Aramis in Ethiopia in 1994 changed all that in the eyes of its discoverers. And in 2009, they let the rest of the world know about it, and through an entire issue of Science devoted to this fossil and other fossils of the same animal recovered from the same general area. Their analysis concluded that the Ardipithecus fossils showed that human beings had, in fact, never passed through an ape-like phase in our evolutionary career, that the last common ancestor of human beings and chimpanzees had been a quadrupedal animal that moved around in a rather monkey-like fashion in the trees, and that the immediate ancestors of our own bipedal kind of animal, beginning with the Australopithecus material from eastern and southern Africa in the Pliocene and Pleistocene, the immediate ancestors of those bipedal apes had been animals that were not arm swingers, that were clambering quadrupeds in the trees and upright bipeds on the ground. And that the best example of that was the Ardipithecus ramidus material. So today we've brought here two very distinguished senior scientists, primary world-renowned experts on human evolution and the morphology of human fossils, to talk about their changing perceptions of Ardipithecus and what kind of effect the Ardipithecus discoveries have had on their overall interpretation of the early phases of human evolution. So I'd like to begin our discussion by handing over the podium to Professor William Kimbel, who is the director of the Institute of Human Origins at Arizona State University, who will tell us about the Ardipithecus finds, place them in their phylogenetic and evolutionary context, and then go on to tell us about the skull of Ardipithecus and its dentition and what sort of difference that makes for our understanding of early human evolution. Bill?
Thank you very much, Matt, and I appreciate the invitation to participate in these BU Dialogues in Anthropology with my good friend and colleague, Bill Jungers. And the essential message that I will bring today is that with respect to Ardipithecus ramidus, despite its amazing and in some senses unanticipated constellation of anatomical features, it is a species that is indeed more closely related to us than it is to a chimpanzee. And that the cranial base, the base of the skull, constitutes some of the strongest evidence for this conclusion. The work that I'm going to talk about today stems from a collaboration I've begun with colleagues Dr. Gensua, Tim White, Yoel Rak, and Berhane Asfaw, and leverages work that was done in the early 1990s by this group on some of the first discoveries of Ardipithecus that became the holotype specimen, the type specimen of the species. To set the context, this is a phylogeny of African hominoids showing the relationship between gorillas, chimpanzees, and humans. The key question we want to ask about this phylogeny, in particular, the point at which the chimpanzee and human lineages diverged, is what changed there? There are a lot of characteristics, a long catalog of characteristics in which humans are different from chimps and other African apes. And the one question we want to know is, from upright posture all the way to lithic technology and sophisticated communication, when did these characteristics change? When, where, and how did we become unique? In order to answer that question, we must rely on the fossil record. And it was only within the past 20 years or so that the fossil record of human evolution became populated by fossil assemblages that bear on the time when we last shared an ancestor with the chimpanzee between 6 and 8 million years ago. Before that time, our earliest representatives were Lucy and other early Australopithecus species that took us back only 3 to 3.5 million years ago. And so these fossils that have been found fairly recently fill a tremendous gap in the time scale of our evolution and hence in our understanding of it. The best known from the point of view of skeletal evidence is, of course, Ardipithecus ramidus. This is a species that was found by Dr. Tim White's team in the Middle Awash research area of the Ethiopian Rift Valley and has, as Matt suggested, really turned on its head some of our preconceptions about the earliest phases of human evolution. Ardipithecus ramidus raises a number of key questions. The first of which is it in fact more closely related to us than it is to a chimpanzee or other apes? The second is, if it is in fact more closely related to us, how? Is it a linear ancestor of later humans, or is it a side or parallel branch? And the third is, to the extent that Ardipithecus ramidus departs from the anatomy of humans and our closest living relatives, the chimpanzees, what does it say about the expected anatomy in the last common ancestor of chimps and humans? I can't address all those questions today, and my colleague and friend Bill Jungers will do so. I'm going to focus on the first one, and principally from the point of view of the skull. The best preserved specimen of Ardipithecus ramidus, and this is a generous description because at its finding it was in terrible shape and required a great deal of effort to clean and reconstruct and understand, is the specimen ARA VP6/500, which is a partial skeleton that includes teeth, part of a lower jaw, and very badly crushed elements of the skull. The first inferences as to the human-like status of Ardipithecus came from one of the specimens that was found early on in the study of Ardipithecus, ARVP1/500, which is an undistorted skull base consisting of two components of the temporal and occipital bones. Most of the inferences that I'll be talking about today come from this specimen. But in addition, there are characteristics of the canine teeth, a set of characteristics that also support the idea of a human relative status for Ardipithecus that I will briefly review as well. Now, this is an image that shows the reconstructed skull of the 6/500 skeleton, juxtaposed with the temporal and occipital bones from the undistorted specimen from the year before, the 1/500 specimen. And this is a reconstruction done by Gensua using scanned digital images and published by him and his colleagues in the journal Science in 2009. And here it's compared with the skull of a chimpanzee and a human. Right off the bat, and in a very superficial, almost gestalt sense, we can see that the skull of Ardipithecus is indeed very ape-like in a generic sense. A very tiny brain, first of all, of only about 300 cubic centimeters, actually smaller by a little bit than the average for chimps, certainly much smaller than what we see in humans, and hence the brain case is very low and small, coupled to a face that indeed has an ape-like aspect to it. You can see that the face, the maxilla, and the snout project downward and outward from the brain case. But as I will now show, these are superficial similarities that belie some other interesting differences that in part, in comparison to chimpanzees, can be tied to the canine teeth. So here we see the canines of a chimpanzee, male individual, and is well known, even in very advanced stages of wear, old adults. The chimpanzees and the canines in chimpanzees are long and interlocking teeth. And of course, this relates to their lifetime of sharpening and honing in a polygynous social system. When we move to humans, naturally, we see that as individuals grow, the canines wear down from their tips so that their wear planes are essentially in line with those of all the other teeth. Now, down below here, from Gen Sua's article in Science on the dentition of Ardipithecus, we can see 6 upper canines of Ardipithecus in different stages of wear, from barely worn, a relatively young individual, all the way to a very old individual. And you can see, as you move from left to right, the wear on those canines is principally from the tip. That is, as the individuals are growing older, they are wearing their canines more like humans than they are like apes. This, it seems to me, and supporting the work of Sua and others, is one sign of a close phylogenetic relationship between Ardipithecus and humans to the exclusion of chimpanzees and other apes. Now, if we move to the cranium itself, this is a superimposed image of the Ardipithecus cranium on that of a chimpanzee. And in comparison of the two, you can see that unlike the chimpanzee with its tremendous projection of the snout beneath the nose and relatively deep face, the face of Ardipithecus, despite its prognathism, is actually less projecting and less vertically deep than in an African ape. One of the other differences relates to the position of the occipital condyle, which is where the cervical vertebral column articulates with the base of the skull. And in Ardipithecus, the occipital condyle sits almost right underneath the auditory opening, the external auditory meatus, whereas in chimpanzees, the occipital condyle is in a much more posterior or rearward position, and we'll return to this in a little bit. One of the other differences relates to the area where the chewing muscles, the temporalis on the brain case, intersect with the neck muscles that support the head on the cervical vertebral column. And in humans and all other hominins, including Australopithecus, that junction is relatively low and horizontal on the back of the skull. If we move Ardi aside and take a look at a chimpanzee, we can see that that junction is much more steeply angled because chimpanzees, being primarily quadrupeds, have that head slung out in front of a relatively horizontal cervical vertebral column, and the neck muscles reach way up on the back of the skull to support it. A very pronounced difference between Ardipithecus and a chimpanzee. In this image, we see a bisected images of a chimpanzee skull on the left and Ardipithecus ramidus on the right. There are 2 key differences I'll draw your attention to. One is the extremely pronounced and high supraorbital torus of the chimpanzee, which Ardipithecus lacks. In chimps and gorillas, uniquely among the hominoids, you get this tremendous development of the supraorbital torus that tends to obscure the brain case in facial view, not in Ardi. Similarly, look at the great depth of the maxilla in chimpanzees, as this whole snout area is quite a bit more extensive than in Ardipithecus. To some extent, this traces to the very large size of the canines and canine roots in chimpanzee, in contrast to the relatively diminutive canines and their roots in Ardipithecus. On the other hand, we can look at the very undeveloped infraorbital plate. This is the area of the zygomatic bone and maxilla right beneath the orbit, which is quite shallow in chimpanzees, similarly shallow in Ardipithecus. So this is a primitive ape-like retention in the Ardipithecus face, and that's reinforced by the fact that both in Ardipithecus and in chimpanzees and other African hominoids, the zygomatic arch, which carries the masseter, the other major chewing muscle, out alongside the brain case, is tucked in. It's not a flaring arch as we see in some later hominins, such as Australopithecus afarensis. And here we see two differences between Ardipithecus and early Australopithecus, one of them being the tremendous depth of the infraorbital region beneath the orbit here in Australopithecus compared to Ardipithecus, and note the great flare of the zygomatic out around the side of the brain case compared to the narrow, tucked-in zygomatic of Ardipithecus. These changes most likely reflect in Australopithecus early steps toward specialization of the chewing apparatus, which is distinctive of this entire grade of early hominins and is not present in Ardipithecus. Now, most of the inferences on the cranial base, as I mentioned, come from this specimen found in 1993. It's one of the holotype specimen of Ardipithecus ramidus, ARA VP1/500. Two pieces of the cranial base. There is the midline present on the basioccipital component, so we have good control over the position of bilateral landmarks. This was the specimen on which the first inferences were made by Tim White's team back in 1994 on a relatively short cranial base with an anterior foramen magnum and occipital condyle. We have taken that initial analysis, have examined it, and moved forward, and the results I'll now share with you, in my opinion, make the cranial base of Ardipithecus even more similar to you and me as compared to a chimpanzee. Here we see the anterior point on the foramen magnum, and here we see the two openings of the carotid canals on the cranial base, and it is generally true in humans and other hominins, these landmarks align in very similar coronal plane, okay? So that's where we're going to start, and we'll take the analysis one step further. Now, in this slide, we have images, drawings of a gorilla skull on the left and a human skull on the right. You can see very clearly the difference in the position of the foramen magnum on the skull base, it being relatively posterior or rearward in the gorilla, much further forward on the skull base in the humans. We can render this difference graphically using rectangles and anchor the rectangles on the anterior point of the foramen magnum, called basion, and then run that rectangle forward to these holes here called the foramina ovale, just passage for one of the cranial nerves here. And you'll note that in comparison to humans, in apes, that rectangle it describes a square almost, equal on all sides. Whereas in humans, the foramen magnum has moved forward, in essence, spreading out these neurovascular canals along the cranial base, and the rectangle is indeed elongated and short from front to back. The fact that the human cranial base is so short should not come as a big surprise to us when we look at data expressing the length of the cranial base in great apes, shown here from gorillas on down to bonobos or pygmy chimpanzees, all of which basically have a cranial base length expected for their skull size, as expressed here by the breadth between the outer margins of the orbits, the so-called biorbital breadth. Australopithecus already shows a human-like shortening of the cranial base, according to these data, with two specimens of Australopithecus afarensis from Hadar falling well below the main cluster of apes seen above. The question is, how does Ardipithecus fall into this relationship? Well, we don't have the breadth between the orbits on the 1/500 skull, but we can start with our rectangle. And if we use the anterior point on the foramen magnum as our anchor point, and some of the other landmarks preserved or projected onto the base, we can see that as in humans, the rectangle is wide and short, in contrast to the elongated, more square shape of the rectangle in the great apes. So that's one indication that not only is the cranial base short, but it's also wide in Ardipithecus as in humans. And here are some metrical data to support this conclusion. We had to go into, to get these data, we had to make some estimates of cranial base length. I can certainly go into those in more detail in a question period. We don't have time to go into it today, but here are a range of estimates of cranial base length showing in relation to a different skull size surrogate, the width between the auditory canals, showing that Ardipithecus ramidus falls with humans in having a relatively short cranial base compared to the great apes. And if we put now an estimate of cranial base, or an actual measure of cranial base width into our equation, we can see that Ardipithecus, indeed in both length and width, falls in with modern humans. Great apes with their long, narrow bases, humans with their short, wide bases. Here's a couple of Australopithecus skulls, and Ardipithecus falling with those. Now, there have been two main explanations for these kinds of changes in the cranial base in human evolution. One of them posits that the foramen magnum moved forward and the cranial base got short when humans stood up, and the vertebral column, the cervical vertebral column, moved forward on the skull base in consequence, as a comparison to the situation in the quadrupedal great apes. And so when we look at skulls in cross-section, we can see, for example, that the cervical vertebral column exits or articulates with the back part of the cranium in the gorilla and other apes. In humans, on the other hand, it is much more centrally located, as we've seen, and descends vertically. The question is this a locomotor change? However, it's not the only change that occurs in the skull and in the skull of these apes. Another difference has to do with brain size. So, for example, the human brain is many times larger than that of an ape, and the most efficient way to pack increased volume in a given space is as a sphere. And the notion that over time, the more spherical, enlarged brain of humans essentially altered the shape of the back of the brain, bringing the foramen magnum forward on the skull base in humans. So the question is, what is responsible for these changes? Is it bipedality or is it brain size? I would suggest that given the 300 cubic centimeter brain size of Ardipithecus, locomotion is probably a more likely functional tie to some of these cranial base changes, speaking for myself. What Ardipithecus shows us, however, that bipedality, as my colleague will discuss, was an early change shortly after the split of the chimpanzee and human lineages. That cranial base structure, such as the position of the foramen magnum, if it is indeed tied to locomotion, as I would think, possibly reinforces the notion that Ardipithecus was an orthograde biped, and early change in canine structure as well, which is probably tied to social structure. With Australopithecus, we get a shift to more obligate terrestrial bipedality, and also, possibly related to that, a specialization in the masticatory system, which we already see developed in Lucy and other Australopithecus. And then, of course, it's not until the origin of Homo, the elaboration of our own genus and its lineage, we see an increase in cognitive ability, possibly tied to more sophisticated lithic technologies as a means of dealing with resource utilization. So, in conclusion, Ardipithecus cranial base structure ties that species to us. It is an unusual creature, as you'll hear more about in a minute, to be sure, but its cranial base structure speaks unequivocally, in my view, to a tie to the human species. And that structure, therefore, in the central cranial base has been with us for at least 4 million years. And in my view, it is more likely to be related to body posture and locomotion than to brain size, given the very tiny brain of Ardipithecus. I thank you very much.
Thank you, Bill. That was very forceful and very persuasive. And your conclusion, which is that the cranial shape of and cranial-based proportions of Ardipithecus are giving us a oblique signal of bipedality, of bipedal upright posture and locomotion in this animal, leads us very nicely into the next speaker, Professor William Jungers, who is the chair of the Department of Anatomical Sciences at Stony Brook University, a long-time and well-recognized authority and expert on the evolution and anatomy of the postcranial skeleton, who is going to come up here and tell us about how the Ardipithecus material transformed his own perception of what human evolution was like.
Thank you, Matt. Good afternoon. Again, I'd like to thank you all for your hospitality and for the opportunity to participate in your dialogue series on a subject that's become very near and dear to me over the last few years. As you'll soon see, a dialogue of sorts about Ardipithecus has been going on inside my own head for some time now, and my thinking has evolved and continues to evolve as I learn more. I also find myself today to be in the awkward position of being in almost total agreement with my friend and colleague, Bill Kimbel. I guess it had to happen eventually by chance alone. Here's a much simplified roster of human relatives and possible ancestors through time, not unlike the one that Bill showed, but without any attempts to connect the dots. I would draw your attention to the extinct species in the red oval seen over here in the lower right. These are what I would argue are the best candidates for the earliest members of our lineage or hominid clade, but some skeptics might argue that they are really more pretenders than contenders. Ironically, other experts would argue that they are indeed hominins and probably belong in the same genus. As someone interested primarily in the evolution and functional anatomy of the postcranial skeleton, I've despaired over the complete lack of published postcrania in the case of Sahelanthropus, and by the very scrappy skeletal record of the other two genera, Auroran and Ardipithecus. Now, if bipedalism is a defining hallmark, perhaps the hallmark of our clade, then the collection of fragmentary fossils before 2009 was suggestive, if not exactly definitive. Fence sitting seemed a reasonable wait-and-see position under these circumstances, and wait we all did. That changed rather dramatically in 2009 when Ardipithecus ramidus exploded across the pages of Science, all 377 pages. And I'm still trying to digest all the information presented by White, Lovejoy, and Sua, and many others in this one giant bolus. Suddenly, we had a treasure trove of postcranials, including the incredible skeleton seen on the cover, ARA VP6/500. Some prominent scientists, like Alan Walker, proclaimed Ardipithecus to be the most important fossil hominin ever discovered. And Ardi was everywhere, on the cover of magazines, on television shows, and all over the web. But as her discoverers and describers noted, here was a hominid unlike anything we'd ever seen before, a marvelous mosaic of primitive and derived features, a combination that probably no one would have ever predicted. It was an animal that was a terrestrial biped, but just as comfortable in the trees. It had solved the arboreal terrestrial problem in a manner decidedly unlike chimpanzees. If anything, it was the anti-chimp, and the fossil record was again playing trickster. As a postcranial person, I was stunned by the bounty of elements rarely seen in early hominins, especially the hand and foot bones. There had been some earlier hints that Ardi's postcranial skeleton was full of surprises. For example, Tim White is said to have quipped, if you wanted to find something that moved like these things, you'd have to go to the bar scene in Star Wars. That's provocative, intriguing, but not very helpful. Of course, even with this new mountain of evidence, not everyone was convinced that Ardi belonged to our family tree. A couple of brief exchanges appeared in Science. Some folks felt chimpanzees had been disparaged. And in 2011, this critique appeared in Nature by Wood and Harrison. My two-word summary of their cautionary take-home message is shown here in red: beware homoplazy. In other words, perhaps some of the features heralded as hominine were not due to shared heritage after all, but were merely convergences or parallelisms that could be observed elsewhere in the fossil record and would therefore not serve as a ticket into the hominine club. They presented the late Miocene insular fossil ape from Italy, Ardipithecus, seen over here, as a prime example. And they placed Ardi, Auroran, and Sahelanthropus in this provisional hominoid taxonomy as inserte cetus, of uncertain placement, but notably outside of other hominines. I personally think that Ardipithecus is something of a red herring in this debate, and I'd be happy to expand upon that if anyone is interested. But there were also other skeptics, including this guy, Jungers, who in 2009 said, this is a fascinating skeleton, but based on what they present, the evidence for bipedality is limited at best. He went on elsewhere to say that we have to abandon bipedality as a hallmark of being a hominine sensu stricto if we hope to keep Ardi in our clade. If we had just found the fossils below the neck, it's possible we wouldn't be talking about Ardi as a hominid at all. If we could turn back time. What I'd like to do in the next few minutes is to invite you to join me as I feast upon some crow I cooked up for myself back then. Today, I'd like to defend the following propositions: that Ardipithecus was a facultative biped, comfortable on the ground and in the trees; that Ardipithecus ramidus probably really was an early hominin; and that despite these two factors, Ardipithecus was still a versatile arborealist. So what changed my mind? What precipitated this conversion experience? Well, for starters, Tim White made me an offer I couldn't refuse. He invited me to study the Ardi fossils at the National Museum of Ethiopia after looking at high-resolution casts in his lab in Berkeley in 2010. He allowed me to assemble a team of experts on the hands and feet, seen here with Tim and Dr. Bahani Asfal back at the museum in 2011. I've dubbed this group Team Chiridia because of their expertise in hands and feet. And it includes some very, very well-informed young investigators: Matt Tocheri at the Smithsonian, Kaylee Orr, Sergio Almathia, and Biren Patel. We were allowed to study everything in ATIS, and Ginsua provided access to invaluable micro-CT images for our 3D analyses. Dr. Kimbel also kindly allowed us to study new fossils of Australopithecus afarensis that were unpublished at the time. Acquiring and analyzing 3D models of bones is hard work and time-consuming. We're still in the midst of data collection from laser scanning and CT scanning, so our results to date are preliminary and best seen as a progress report. And our bias remains: it's best to quantify everything you possibly can. So now let me offer 3 lines of evidence that convinced me that Ardi was indeed a biped, a facultative biped that was at home on the ground and in the trees. Here you see the foot of Ardipithecus to the left. This is from the sole or plantar view. Other views can be seen in smaller frames down below. The feature that really jumps out is the divergent hallux, or big toe. It's a mobile, grasping organ that is well suited for locomotion into and within the trees. In other words, it's not adducted and in line with the other toes as in humans and other early bipedal hominins. The lateral toe bones, including the proximal phalanges, are also pretty long and quite curved. And at first I couldn't get past this grasping complex to see the bipedal signature. But let me draw your attention to the upper right panel. What you see here is the third metatarsal of a human, of a chimpanzee, and another individual of Ardipithecus that preserves all the anatomy of this bone. The shaft of the human in Ardipithecus is quite straight. It's more curved than the chimpanzee. But I want to point out especially this little bump right here. This is known as the dorsal dome. This dorsal doming reflects an extension of the articular surface for the base of the phalanx to ride up onto the dome during the extreme extension that occurs during the push-off of walking. The bases of the phalanges that articulate with the dome were also angled or dorsally canted to facilitate this toe-off movement. We agree with Lovejoy and colleagues that this joint complex is derived and occurs only in bipeds. It's been a difficult feature to try to quantify, though. And we think we've come across a promising method called 3D geometric morphometrics for this purpose. Using XYZ coordinate data and a method called procrusty superimposition, you can now see a mesh of points that are derived from these landmarks that are superimposed over the articular dome itself. And what we've done is summarized the information in this kind of analysis in sort of two primary axes of variation. And what you see here is a group of apes and monkeys separated completely from modern humans. And we believe that this 3D shape information is really being driven by the doming. And we fully expect Ardipithecus to fall into this human envelope when it's included along with other extinct hominins. The second anatomical complex, the same doming canting complex has been used recently to identify a 3.5 million-year-old partial foot from Bertelli in Ethiopia. This was a previously unknown bipedal hominin with morphological affinities to Artie's foot. It's been superimposed on a silhouette of a gorilla foot here. This shows the same complex, dorsal doming and canting. And it is very, very different than its contemporary Australopithecus afarensis. It suggests that Ardipithecus-like adaptation for both arboreality and terrestrial bipedality was not even necessarily transitional, but instead a long-lived and very successful evolutionary strategy that persisted in Ethiopia. We were also trying to characterize the shape of individual foot bones, like the talus seen here, that bone that connects the rest of the foot to your leg at the ankle. Using methods developed by Matt Tucheri and Kalior, we've segmented out articular versus non-articular parts of the bone and calculated angles between joint surfaces and degrees of curvature of these surfaces. Artie's talus is seen up here in the black box. A gorilla talus is shown over here to the right, showing you in different colors how the different surfaces have been segmented out. Now, in combination, all these features allow one to discriminate quite clearly among different species, here indicated by different polygons. Here are humans over here, gorillas, chimpanzees, orangutans, and some Miocene apes called proconsul, and baboons are over here. Artie is seen right here in the middle, in the one primary axis overlapping with this group that's fairly arboreal, to be sure. But along the second axis, it's moved down towards humans and gorillas. So it's in an intermediate shape space that is shared by a lot of other fossil hominins that we all agree are bipedal. And if you were to lump all the other fossils into a group as a separate category, and then classify Artie by itself, it is classified as an early hominid and not like anything else. Now, eventually, we hope to put this information from all the individual bones of the foot back into an integrated whole. There we go. The second anatomical complex that indicates bipedality is the mosaic anatomy of the bony pelvis. Yes, this bone is badly crushed and has required extensive reconstruction. Tim White jokingly refers to this as the Irish stew. But I can confirm that informative parts are indeed intact and require no reconstruction, including the lower half of the iliac blade, the area between the hip joint and the sacroiliac joint seen between these two red horizontal lines. This is also known as lower iliac height. As in humans and later fossil hominins, this distance is very short in Artie. and indicates that the center of gravity has been relocated closer to the hip joint in a more stable position for a biped. Again, we see a mosaic anatomy. Here's Ardipithecus compared to Lucy, a modern human, and a chimpanzee. There are other features that hint at a hominin-like morphology, including the incipient sciatic notch. It's as if we had a hominin false pelvis grafted onto a more primitive, ape-like true pelvis. In addition, if this is now showing Artie in gray, superimposed on the pelvis of Lucy. If this reconstruction is also accurate, it actually is surprising because it places the lesser gluteal muscles, those muscles used in lateral balance, in a more favorable position than even we see in Lucy. The third and final feature of the skeleton that I believe signals bipedality isn't even part of the postcranium. It's the anterior position of the foramen magnum that Dr. Kimbel talked about so eloquently earlier. This new study by Rousseau and Kirk provides independent and, I believe, persuasive evidence for not just orthogrady or erect posture in Artie, but for bipedalism specifically. They examined bipedal and quadrupedal marsupials and rodents in addition to primates and discovered a predictable relationship that to them suggests that foramen magnum position may be used to identify bipedal adaptations in fossil hominins. Accordingly, the anteriorly positioned foramen magnum of Sahelanthropus and Ardipithecus compared with extant hominoids provides strong evidence that both fossil genera were indeed habitually bipedal. Artie, along with Sahelanthropus, was described as a cladistic hominin even before 2009, a finding I conveniently forgot about in my earlier skeptical remarks. This phylogenetic study by Straight and my Stony Brook colleague, Fred Grein, had used cranial dental characters to place Artie firmly within a monophyletic clade of hominids back in 2004. The derived features seen in the postcranium of Artie linked to bipedalism would only serve to strengthen this phylogenetic hypothesis. The addition of primitive characters really would not matter. Now, so in other words, here's Artie nested between Australopithecus anamensis and Sahelanthropus. This kind of analysis doesn't tell us if Ardipithecus is ancestral to anything else, but it does suggest it's a hominin. The reassembled hand of Ardipithecus is also fascinating. Many aspects of its morphology are similar to the same bones in Australopithecines, including some of the wrist bones, the metacarpals, and the phalanges. It's a powerful grasping organ, but it's not especially long. It's well suited for grasping in a variety of arboreal activities, but it is not the elongated hand of a specialized suspensory ape like the gibbon, chimpanzee, or orangutan. It's also not the specialized hand of a knuckle walker, nor is it the specialized hand of a digitigrade quadruped or of a palmigrade quadruped for that matter. This is a slide that has a lot of information, but I just want to point out two things. These are schematics of the thumb and the fourth ray, and these are the individual elements of those bones. When adjusted for overall body size, Artie's hand proportions cluster it over here with gorillas, humans, Australopithecus, and proconsul, and away from the highly suspensory elongated digits that we see and hands that we see in suspensory apes. However, the thumb of Artie is quite short relative to the fourth digit, pretty much ruling out the pad-to-pad precision grip characteristic of humans and probably Acetiba. Now, no presentation would be complete without a comment on phalangeal curvature, at least one from Stony Brook. Artie's proximal and middle phalanges are very curved, more so than in a chimpanzee. Curved phalanges are biomechanically linked to arboreality, even when controlling for phylogeny. When estimated as the included angle seen here, the curvature of Artie's proximal phalanges, plotted here in this plot, exceeds that of chimpanzees and bonobos and approaches that seen in some siamang and spider monkeys. This is a powerful arboreal hand. If I were only of these elements alone, I might even think it is a suspensory hand. Now, this is just a teaser. I'll wrap up quickly, just to show you that 3D geobium is also useful for looking at some of these small, complicated bones of the wrist. This is the hamate of a chimpanzee. It's also possible to impose a phylogeny onto this and to examine shape, space, and phylogeny simultaneously. And you can see we're getting a good separation of lesser apes, great apes, and humans, and all monkeys. I think this is a very, very promising methodology. And in fact, we just published a paper recently on Auroran, one of the other candidates, that indicates using this methodology, in this case on the proximal femur, that the femur of Auroran tugenensis exhibits morphometric affinities with both Miocene apes, like proconsul in particular, and later hominins. And by putting on the phylogeny, we can see this is one of the cases where the extant great apes are highly derived. Their shape is moved far away from what you expect in the last common ancestor. And this is one of the approaches we'll be taking in looking at other elements. So some conclusions. Ardipithecus probably was a biped, almost certainly was a biped, when on the ground and perhaps in the trees. If the ancestral condition was pronegrady, that pronegrade signal is relatively weak, almost extinguished, I think. I think Ardipithecus was a versatile arborealist and capable of climbing with a climber with grasping hands and feet, but was not a suspensory specialist. I think Ardipithecus and chimpanzees solved the arboreal-terrestrial challenge in very different ways. And the Ardipithecus is probably a bona fide basal hominid and related somehow to the Australopith Homo lineages. And finally, and more as a memo to myself, it's not a bad idea to have a look at the fossils before making proclamations. Thank you very much.
Thanks very much, Bill. We appreciate your coming here and sharing your expertise and your knowledge and your new understanding, and especially your crow eating with everybody here at BU and everybody out there on the web. And in the midst of this harmony of sweet concord that we're now hearing from previously somewhat opposed viewpoints, I guess it falls to me to inject a note of discord into the proceedings by raising some questions about other aspects of the synthetic view of Ardipithecus that emerged in the 2009 papers and that has become more and more widely accepted among people like yourselves. Because I have some problems with some of this. And let me just begin by saying that people who are not accustomed to thinking in evolutionary terms and haven't had a lot of training in comparative anatomy often look at presentations like the two that we've just had and say to themselves, oh, wait a minute. That's clearly the head of an ape. It's got a little tiny brain and a big protruding face. And how come a few little features like the exact position of the carotid artery canals or that little wear facet on the back of the upper canine outweigh this obviously ape-like gestalt of the head? And similarly, when you look at the Ardipithecus foot, yeah, okay, so it's got some features, that little bump at the top of the distal ends of the metatarsals. But look at the thing. It's got long toes, it's got curved phalanges, and most importantly, it's got a big thumb-like opposed first toe that's tremendously primate-like. How can you possibly decide that this thing is related to a human being when it doesn't resemble human beings? And the answer is pretty simple, that not all resemblances count. The ones that don't count are the ones that are primitive, that you have reason to believe that were inherited from some ancestor way back in the past, before the last common ancestor of the two animals or the three animals that you're trying to look at. Okay, for example, if we look at human beings and opossums and hippopotamuses, the fact that human beings and opossums have hair all over their bodies and hippopotamuses don't is not a reason for suspecting that people are more closely related to possums than they are to hippos. Okay, because it's a primitive feature. Primitive mammals had hair, and therefore you expect mammals to have hair. When they don't, it's what's called a derived feature or a synapomorphy. And it's only those non-primitive shared features that are reasonably interpreted as being a signal of evolutionary relationship, of close phylogenetic affinities. And with that in mind, I'd like to turn to another aspect of the Ardipithecus story that I continue to have a lot of trouble with. Okay? One of the main reasons for the sort of turnover in opinion about what human ancestors looked like in the mid-20th century was the accumulation of an enormous amount of comparative anatomical evidence beginning with publications by Sir Arthur Keith back in the 1920s, that human beings and all of the extant apes, the great apes, gorillas, chimpanzees, orangutans, and the lesser apes, the gibbons, all shared with human beings a large number of anatomical features that could be reasonably interpreted as having originated as adaptations to moving around in the trees in a suspensory position, hanging by your upper limbs. Okay, and I just want to run through a few of these features. I'm not going to actually talk about them. I'm just going to put up a list here so you can be impressed by the number of lines in the typography. Okay, there are a lot of things about the human postcranial skeleton, about all sorts of aspects of it, the pelvis, the shoulder girdle, the bones of the arms and legs and feet, about the soft anatomy in the viscera, that tell us that we have reason for suspecting that the last common ancestor of all the apes shared these non-primitive, not found in primitive primates, these derived features that originated as adaptations for arm swinging, for upright, orthograde, locomotor postures, and for suspensory adaptations in the forelimb. Features associated with supporting the body weight through the forelimb, under overall tension in the limb. Traits associated with increasing the reach and mobility and power of the forelimb in propelling and suspending and turning the body. Features associated with the reduction of adaptations for quadrupedal running and leaping, like the reduction of the deep back muscles and the shortening of the vertebral column between the pelvis and the rib cage. And visceral traits associated with this 90-degree shift in the gravity vector from running at right angles to the spine to running parallel to the spine, so that our guts tend to fall out at the bottom through our pelvic opening, and we have adaptations to prevent that sort of thing from happening. We shared all of these adaptations with the living apes, and they're not seen in any quadrupedal monkeys. And given that fact, it's hard for me, at least, to be entirely persuaded that these things originated again and again and again in many, many different lines of animals leading to the living great apes and humans and the lesser apes, which is required if, as the discoverers and describers of Ardipithecus insist, the last common ancestor of humans and chimpanzees was an above-branch quadruped that had no suspensory adaptations and didn't hang by its upper limbs. Okay, so I'd like to throw that particular dead fish out on the table here in the middle of the ice cream party and ask our distinguished visitors how they would respond to that kind of critique.
You raise, I think, a variety of very good points, Matt. I think one of the problems is that if we go to the fossil record, it doesn't speak very clearly to when those events happened. And some of the events in your list are not clearly linked to suspensory behavior at all, like loss of the tail. But you may have noticed that I sort of avoided talking very much about what the last common ancestor of chimps and Ardipithecus and early hominins looked like, of how Ardipithecus might fit into that profile. I know that the last common ancestor has been characterized as a pronagrade animal, and I think modeled in large part on the Miocene apes, earlier Miocene apes of Africa. And it's true, with the rare exception of maybe Ardipithecus, none of those really show orthograde anatomies. So I'm not disputing the possibility that there was more suspension involved than maybe has been characterized in the pages of science. I think that it's probably the case that with a hand and a body design like Ardipithecus, that it was capable of almost any kind of arboreal behavior except pronagrade activities.
The only thing I would add to that, and I do agree with the idea that the fossil record is the ultimate arbiter of the chronology of acquisition of these characteristics. The very thorough list you showed, Matt, is based on comparison of living African apes with humans. But the other thing is that the extent of convergent evolution or homoplasy here is and it's important for the audience to understand this, is not dependent on Ardipithecus being uniquely related to Australopithecus and Homo. That list is more or less the same if it is more closely related to chimps than to humans, or to gorillas than to chimps and humans. Many of those characteristics remain convergent. If indeed Ardipithecus represents the primitive bow plan for the body that was ancestral to all hominoids. So.
Okay. We've had some questions sent in over the internet and by email. We have time, I think, to ask a couple of them. Let me begin by asking a question about the foot skeleton from Bertelli that Bill Jungers showed us. This is coming from about 3.5 million years ago. Right? And up until the discovery of that foot, there was a pretty general consensus that most of the, at least Ethiopian material from that time range, and there isn't a lot of other material from that time range, could be put into a single species, namely Australopithecus afarensis. And now we seem to have pretty good evidence that there were at least two animals present in that melange of material that we're calling Australopithecus afarensis, the Lucy species that has been thought to go with the Lucy skeleton. One of which had a grasping, opposable big toe, and the other one didn't. Okay, so I've got a question here sent in by Bernard Wood down at GWU, who wants to know how you go about doing the alpha taxonomy in this 3.5 million year old collection of East African fossils, and whether there is evidence for thinking that any one region, teeth or cranial vault or upper or lower limbs, shows more or less evidence of morphological discontinuity, of homoplasy, or is likely to be a more reliable source of hypotheses about what's related to what and what belongs in which species.
Well, I think the Bertelli foot certainly tells us that there was more than one lineage at 3.5 million years ago. That's quite clear. The dilemma with regard to alpha taxonomy, of course, is that almost all of our diagnostic characteristics come from the skull and dentition. So the key to unraveling the alpha taxonomy in this time period will be to find another Lucy skeleton, for example, in which you have a foot like the Bertelli foot with diagnostic craniodental material. Having said that, however, of course, there's a long history of interpretation of the Hadar remains in particular that has a minority but very important position that has suggested, in fact, that the Hadar sample itself consists of two taxa, one of which typified by Lucy and another which typified by the 333 or first family material. So, it is a viable, more viable alternative than it was before the foot. The question is, how do we operationalize the from here on out? And I don't grant any particular part of the anatomy primacy with respect to phylogenetic value or homoplasy, frankly, because homoplasy to me in phylogenetic reconstruction is not a tool for analysis, it's an outcome of analysis.
Mm-hmm. So the question is, which heap of purported synapomorphies is the biggest heap?
Well, clearly right now it's from the, synapomorphies we're not dealing with at the alpha taxonomic level. We're dealing merely with phonetic differences, and right now they're mostly craniodental, of course.
And to the credit of the people who described the Bertelli foot, they made no claims as to what species it belonged with. It was only diagnosed as a hominin based on that one, and I think rather powerful complex of domine and canting. So I think they've left the door open as to what that might ultimately belong to.
There are a lot of interesting questions to be asked here. We hope that you will come and help us ask them and maybe listen to some of the answers that you get from our distinguished guests and other members of our roundtable panel this afternoon at 4:30 here on the 4th floor of Hillel House on the BU Charles River campus. We'll be having an open public panel discussion featuring Professors Kimbel and Jungers and some of the BU faculty, asking and answering pointed questions about Ardipithecus and its meaning and how it fits into our understanding of human evolution. So please come along, bring your questions, listen to some of the other questions that we've got to ask here and that I know all of the participants will want to direct to each other, and join us afterwards for a reception and for a chance to talk one-on-one, up close, to the participants in this 8th iteration of the BU Dialogues in Biological Anthropology.